Okay, good morning, students. So I guess most of the students have joined, and we can start with the class. And in the meantime, others can join, right? So let's just quickly go through the previous lecture that we took last time, which was basically about we discussed about the differences between viruses as a pathogen, bacteria as a pathogen, and fungal pathogen. So what are the viruses we have been discussing already, right, since last week. So we discussed about the bacteria also, that how bacterial pathogens they differ from the viruses, and what are the diseases they cause, and what are the management aspects there, and few important diseases of in plants that bacteria cause and in humans they cause. Then we discuss about the fungal diseases also and their management, and the diseases that they're causing in humans and the plants. So my first question is for Ayushi Jan. Yes, Ayushi, can you just tell me what is the major difference between bacterial pathogens and viral pathogens, or a bacteria as a pathogen or a virus as a pathogen?
Ma'am, like viruses, they are not living organisms and they can only reproduce inside the host cell. And
Yes.
if they are found outside the host cell, they are usually inactive or dormant. Therefore,
Mhm.
their life is requires the hijacking of the biochemical activities of the living cell. But whereas if we talk about the bacteria, it is a living organism and it consists of a single cell. They can
Mhm.
generate energy, they can make their own food, and they can reproduce by binary fission, which allows them to live in different places like soil, water, plants, or even it can live in inside human body.
Fine, good, Ayushi. My next question is for Nehal. Nehal, can you just tell me, can you just name two diseases which are caused by bacteria in humans, or just any one disease? Okay. Yes, Sanjana, this question passes on to you. Can you please name any disease which is caused by
Yes, ma'am.
viruses in human? Not virus,
Ma'am,
bacterial
virus.
pathogen. Yes.
Okay, ma'am. Tuberculosis and pneumonia.
Yes, these are the two important diseases caused by the bacteria. And then Tanvi, can you just tell me important bacterial disease in plants? Anyone? No? Anyone?
Ma'am, blight of beans or like rotting.
Yeah, see, on the basis of symptoms, you can say there's a lot of leaf spots caused by bacteria. There are a lot of rotting and scabs caused by bacteria. There are a lot of bacterial wilts caused by maybe fusarium wilts or other wilts, pseudomonas or any other bacterial wilts also. Bacterial wilt, there's a bacterial wilt, there's a fungal wilt also. So there are a number of diseases. There is a crown gall or galls like diseases which are caused by bacteria in the plants, right? Yes. So can just someone okay, I'll ask names. Okay, Manaswini, can you tell me about the management of fungal plant pathogens, fungus in a plant? Yes, Manaswini, am I audible?
Yes, ma'am, you're audible. Sorry, I was
Yeah,
just having some network issues.
yeah.
So, as far as I know, so as we know, most of the fungal plant diseases, their symptoms are like root rot and blight and all, like most
Mm-hmm.
other plant diseases. So one way to maybe curb it is to continuously check if the plants are in proper condition and to ensure that there is no growth of fungus, and there are antifungal medications which could help. And other than that, just there's also different biological methods to ensure that the fungus doesn't grow. So
Yes.
mostly it's I forgot the word, but fungicidal
Yeah,
chemicals
it
which
is.
could be
I
helped.
discussed about the integrated disease management. I
Yes,
focused,
yes.
I put a lot of focus on the integrated disease management and how integrated disease management is very important in any kind of a plant pathogen. So
Hmm.
that is, see, we discussed yesterday that chemical control is always avoided due to the number of diseases and number of effect it is causing on the humans or animals or even other plants. So to avoid the use of chemicals, we need to look for the other practices, and those practices combinedly they come under integrated disease management. There are biological practices, there are cultural practices, there are like use of a clean seed and a clean planting material. Then you
Hmm.
can just crop rotation, or you just change the time of a sowing in a particular field so that prevent the pathogen attack, right? Okay, fine, good Manaswani. So see, yesterday we discussed about various types of the pathogen. Each pathogen is important, and management of each pathogen is important, whether we are talking about plant pathogen or whether we are talking about human pathogens. We are definitely more concerned about the definitely human pathogens because we are losing lives out there, and we don't want to lose people. So somewhere we are, yes, the human diseases are more important to study, and a lot of work is already going on. People, lot of research and a lot of people are working on that. But apart from that, we need to work on the plant pathogens also, because plants are our source of food. And agriculture, India is an agricultural country. Lot of production, a lot of livelihood of the people is dependent upon the agriculture. And we know the kind of population that we have today, and we are a developing country, we are not a developed country. So we need to feed more number of people, and for that we need to make sure that our agriculture is good. And yes, believe me, it is definitely good. You know, when the green revolution came in India, and a lot of good wheat varieties started coming out. There was a one time when there was a green revolution. Then the time came, there was a white revolution, which is the milk revolution. Now after that, there's a gene revolution. So gene revolution was a common term which was used for the transgenics. When we started moving towards the transgenic, when we thought that the development of a transgenic varieties with good the productivity can be increased, the we can enhance the quality and quantity both. So by use of the transgenic facility, then that part was known as the gene revolution, and we are still going on under that gene revolution stage. How to improve the varieties, how to improve the plants, how to improve the fruits, how to improve all those things, how to improve the resistance of a plants for a particular pathogen. So all those things comes under the gene revolution. Yes. So yes, for any disease or any pathogen, the next step is always diagnosis, and then the next last step is the management. And in human diseases, we always think of the eradication, that how we can prevent this disease from occurring, or how we can just remove this disease from this world. So this is how we are trying right now for the corona. A lot of vaccines are still being developed. A lot of vaccines have been already developed, and we all are getting vaccinated. So this is how we work towards the management of any disease, right? So today's lecture is basically about the origin of viruses. See, maybe 2 days back or in the previous lecture, I told that it is very difficult. We still are. We still cannot say that any hypothesis is short or 100% for origin of any viruses. But still, there are around 3 hypotheses which have been proposed, and there are different theories for these 3 hypotheses, that they tell about the different things, that how viruses may have originated, and how they, so evolution of viruses will try to take up in the next chapter, that how they are being evolved. So to study for the origin of the viruses, let's first there is still a very heated discussion, and still an argument going on, that are viruses alive? So to answer this question, we should first know that what is life, what do you actually mean by life. So there are certain definitions which are being given by the biologist, which there are certain properties which are being considered to be part of the life, that okay, if you have these kind of activities or properties, then we can consider you living, basically. So basically, those who can grow, reproduce, maintain an internal homeostasis. They respond to a stimuli and they carry out various metabolic processes. So all these properties are basically they reflect that that person or that organism or that he is alive. It is it define the life basically, right? Or definitely they evolve. We all evolve from a within a particular time period throughout. Yes, the evolution in the humans is very slow as compared to when we talk about the pathogens. There's another difference for that, but yes, we all are evolving, and from past so many years we have evolved so much. So do viruses confirm this criteria? So answer is yes and answer is no also. We all know that they reproduce. Yes, they replicate, but definitely they reproduce by some way. They are replicating, they are producing their progeny, they are, giving more of their types, giving rise to more of their types. So we can become infected with a just a small number of virus particles by inhaling, maybe through coughs, or maybe transmission by any other mode, and then we become several days later. Because why it is that? Because now virus has replicated his number, so it has replicated to many fold in our bodies, right? So likewise we probably know that, we know that viruses, they evolve over time. Number of strains of a same viruses, number of a new virus types, they are there. So this gives indication that they are evolving also, right? So we know we need to develop some flu vaccines every year, because flu viruses or influenza viruses, they are changing. Lot of changes in their genome are there every year, right? But then viruses, why no? Because viruses, they do not carry out metabolic processes. Most notably, viruses differ from the living organism because they cannot generate ATP's energy, right? ATP, we all generate ATP, we all use that to carry out other kind of processes. They don't do that. They also do not have any proper machinery for the translation. They lack that. They don't have ribosomes. They don't form proteins from independently, proteins from the messenger RNA. So all these somewhere tells that they because of these limitations, they can only replicate within the living host cells, right? So they are obligate intracellular parasites, and according to the stringent definition of life, because they do not have the major characteristics of a living organism, so they are non-living. Not everyone though necessarily agree with this conclusion. Still also, if you try to google out, you'll find lot of arguments in favor and against the fact. So there are many a group of people who still believe that they are living, and another group of people who believe that no, they are not living, right? So this is still going on. So the uh, we know viruses have RNA as genome. Viruses have certain properties which makes them stand out of the league of living forms. Only viruses can have RNA as their genome. Genomes are very small, still they possess functional abilities. They even changed the central dogma of life, right? Central dogma of life is what? DNA to RNA and RNA to proteins. This is how it should be. But now it could be DNA to RNA, RNA to DNA also in some viruses you'll study, and then RNA to protein. So a central dogma of life for viruses is little different. In retroviruses, for example, I'll discuss that example later, right? So with these abilities, they are also known as fossils of RNA world, because their genomes have majorly RNA as their genome, right? So the question again arises that viruses were there before life originated, or viruses originated from within the living organism. So when you study these hypotheses, you'll see that there are three hypotheses, and all three, they contradict with each other, or in some ways they are similar to each other, and a lot of answers are being given there. But still, not one hypothesis, as I said earlier, is sure shot. So we live in a, there are so much in number that we live on a planet of viruses, it is a saying. So there's one statement in 2011, a journalist Carl Zimmer, so there was one book, a planet of viruses. So there they said that we live in a planet of viruses. So now in 2020, this statement seems to be I mean, fine, because the whole world, can you see, the whole world is impacted by a single virus, coronavirus. So definitely their number is more. Definitely they are larger in number, and definitely lot more viruses are yet to be discovered. So this totally is fine that okay, we are living on the world of planet of viruses because they are more in number than the humans. Then a recent study estimates that number of individual viruses on Earth is at 10 quintillion, which is 1 followed by 31 zeros. Can you see the number? They are not visible, but they are replicating at such a fast rate, and their number is more. So that makes them not only the most abundant type of organism on our planet, but there are many more viruses on our world than there are stars in the whole universe. Just a common theme. Sometimes we say, no, stars are enormous, we cannot count them. So, but this is another. Viruses are enormous. We cannot count how many viruses are there. Another recent study placed this overpopulation on a more human scale. Every day, an average of 800 million viruses fall on every square meter of Earth's surface. This is just an, hypothetical statement that this is happening. They are so much in number than every day, so much new viruses, or so much maybe they are multiplying at such a higher rate that they are there. Right? So that they truly are everywhere in every land or a marine ecosystem. दे आर एवरीवेयर, दे आर फाउंड एवरीवेयर. आप एंटार्क्टिका में भी चले जाओगे, वहां पे भी वायरसेस डिस्कवर होंगे. यू गो टू एनी एक्सट्रीम कोल्ड प्लेस, एनी एक्सट्रीम hot place, anywhere, they are everywhere. So everything is impregnated with these tiny organisms whose existence we were unaware until many years, because they cannot be seen with the optical microscope. So they were not known until we were able to see them. So for thousands of years, we knew viruses only from their effects in the sickness and death. So they are known to cause sickness and the death, or earlier as a mystery pathogens. Some call them as a poisons also. So they are there since ages, maybe since we evolved, or maybe before us also they were there. So origin of viruses is very difficult to nail down, because among other reasons they do not form fossils. We can know about, we know the our evolution or the evolution of the plants. How do we know? Because we have fossil records. So we know how they were earlier and how they have evolved. But viruses, they are not forming any kind of a fossil. So all we do is to apply molecular biology techniques to study their genes. And that's how, see, how do we study the differences in the viruses? How do we study they are different? We sequence them, then we do a phylogenetic analysis. That is, we study the evolution of virus. We prepare a phyllo. There are some softwares available where we put the sequences of the viruses, and commands are given, and phylogenetic tree is being formed. So that phylogenetic tree gives us the, the idea about their evolution, from where they have evolved, what was their parents, like the parents, and what was the daughter virus. This is how the terms are being used in the recombination detection program. There are RDP programs which are available to detect the recombination in the viruses. So those are the highest studies that people do later to study the differences in the viruses. But the molecular techniques are basically being used to study the genome of the viruses, how they are evolving, how they are changing, right? So these studies have led to the three main explanation, or the three main hypotheses, which we call. The first hypothesis is the progressive hypothesis, or it is sometimes called as an escapist hypothesis also, right? So according to this hypothesis, viruses originated through a progressive process. Means a step-by process from a basic to the complex one. पहले आप simple थे, फिर उससे step by step आप complex बने. You change to a complex form, right? So they say the mobile genetic elements, or pieces of a genetic material which are capable of moving within the genome. So within time, a certain period of a time, they gain certain ability to exit from one cell to and enter the other cell. So for example, this transposomes, I'm sure you all have heard of the transposomes, or a jumping genes, right? In humans there are around 32% or some percent where we are jumping genes. They keep jumping from one place to the other. So this theory has come from the jumping genes only, right? So this the escapist or progressive hypothesis suggests that viruses originated from RNA and DNA molecule once they escaped from the host cell, and then within a certain period of a time they become independent or a parasitic, obligate parasite, which needed some kind of a host, other host to multiply and replicate. So here it is just a very, I've just tried to take a very simple example of retroviruses. Retroviruses are basically single stranded RNA genome viruses. HIV comes under this, and how they replicate. So when the virus enters the host cell, the viral enzyme, they have an enzyme which is reverse transcriptase, so it convert its a single stranded RNA into double stranded DNA, right? That's why they are called retroviruses. They convert RNA to DNA. Then the viral DNA, it migrates to the nucleus of the host cell, and another viral enzyme, which is called as an integrase, it inserts the newly formed viral DNA into the host cell genomes. Now, the viral genes can then be transcribed and translated. Now, host cells RNA polymerase can produce new copies of a virus single-stranded RNA genome. Now, this progeny viruses or a new viruses, they assemble and exit the cell to begin the process again. So this is how HIV virus or retroviruses, they replicate. They enter the cell and how they multiply. This is what is happening. So they say that this process is very, shows a very close resemblance to the process, the movement of important or though somewhat unusual component of most eukaryotic genome, which are called as a retrotransposons or jumping genes, right? Retrotransposons are mostly found in a eukaryote, and when they are found in a prokaryote, they are called as a transposons. This is the difference, major difference. So these mobile genetic elements, they make up and around 42% of the human genome, and they can move within the genome. They are jumping genes, they are moving from one part to the other part. Now, like retroviruses, these retrotransposons, the viral like retrotransposons, they encode a reverse transcriptase also, and they have an integrase enzyme also. So with these enzymes, these elements can be transcribed to RNA, reverse transcribed to DNA, and then they can integrate to some new location within the genome. So from here, it is being speculated that acquisition of a few structural protein could allow an element to exit the cell. Such kind of an element, they can exit the cell, they can enter a new cell, and thereby they become infectious within a certain period of a time. Altogether, a new organism or a new entity is being formed. So this was basically our progressive hypothesis. That means from basic to the higher. Kaise hua? So this resembles with the transposons, a jumping gene. So they thought that viruses were also like transposomes. So from the transposomes, maybe the retrotransposomes, they have formed a virus-like particles, independent hoge within a certain period of a time and started multiplying wherever they're finding the suitable host. So this is basically the first theory. Then we have a regressive hypothesis. So regressive is just the opposite of the progressive. Regressive में होता है पहले आप complex form में होते हो, and then from a complex form you changes it to the simpler form. पहले आप simple थे, complex थे, with the evolution or with the evolving, you have evolved yourself to such a level that you change to a simpler form. So in contrast to the progressive process, viruses may have originated by regressive or reductive process. So what does microbiologist here argue? They say there's certain kind of a bacteria which are obligate intracellular parasites such as chlamydia and rickettsia. These are gram-negative bacteria which are obligate intracellular parasites. They have evolved from a free living ancestors. उन्होंने वो भी इवॉल्व हुए फ्री लिविंग एंसेस्टर से. So there are certain genomic studies which indicate that mitochondria of eukaryotic cells and rickettsia, they have a common free living ancestor. There are certain papers which indicates this. So it means then existing viruses, they might have evolved from a more complex or possibly a free living organism that lost its some genetic information within over a period of a time, and they adopted a parasitic approach of a replication. So they have evolved from a certain period of a time from a free living organism to the they become a parasitic, and they started replicating whenever they're finding a particular living host or certain kind of a host. No, viruses of one particular group here, which are called as a nucleocytoplasmic large DNA viruses. These are the very large viruses. They are like bacteria. And the largest of the viruses are like Mimi virus is one example here. So they are much bigger than the bacteria. Or example, I'll just tell you that here like they have said that Mimi virus has a genome of around 1.2 million base pairs, whereas polio virus has genome of 7,500 nucleotide base pairs. Mimi viruses are found infecting acanth amoeba, a very different kind of a host basically. So when we study virophages, there we study Mimi viruses. So, but just remember that they are the largest viruses which are being identified till now. So in addition to their large size, these viruses, they are more complex than the viruses, and they have less dependency on the host for their replication as compared to other viruses. For example, they say the pox viruses include large number of viral enzymes and related factors that allow the virus to produce functional messenger RNA within the host cytoplasm. So because of the complexity of the size of these Mimi virus type viruses, DNA viruses, some viruses have hypothesized that these viruses may be descendants of a more complex ancestors. So according to this hypothesis, autonomous organism, initially they have certain kind of a symbiotic relationships they were having. But over the period of a time, this relationship turned parasitic. As one organism, it became more and more dependent upon the other. So as the once free living parasite, they become more dependent on the host. It loses its previously essential genes which are required for the its that free living association for it required. So it loses because it is becoming dependent on some other person or some other microorganism or pathogen. So it does not need those kind of a genes. So eventually it was unable to replicate independently, becoming an obligate intracellular parasite known as virus. So analysis of a giant Mimi virus, basically, this hypothesis is just on the basis of giant Mimi viruses which were discovered. This basically support this hypothesis. But Mimi virus, or these also when they were studying the the Mimi virus, they said that it does not differ appreciably from parasitic bacteria such as Rickettsia. They have certain kind of a resemblance there. So that is how this hypothesis is being proposed. Now, the last hypothesis. The last hypothesis was known as a virus first hypothesis also. So it says, what if virus exists first? Because the both progressive and regressive hypothesis assume that cells, they existed before viruses and then viruses existed, then they came. But this hypothesis said, what if they existed first, or what if they co-evolved with the humans or other life on the earth? So recently there are some investigators that propose that viruses may have been the first replicating identities, even much before than the humans or other form of a life. So there's one study by Kunin and Martin, and they said that viruses existed in a peculiar world as a self-replicating units. Over the time, these units, they become more organized and complex, and they started they started having certain enzymes for the synthesis of membranes and cell wall. So that resulted in the formation of the cells also. So viruses then may have existed before bacteria, archaea, or even all eukaryotes. According to this, they said because they are independent replicating units, they are RNA or DNA independent, and it has been already approved that RNA came first, then the DNA. It evolved first, right? So according to this, they agree that very first replicating molecule consisting of RNA, not DNA. So there's a possibility that they are already there before even the life existed. So we also know that some RNA molecules, ribozymes, they have enzymatic properties, right? So perhaps simple replicating RNA molecules existing before the first cell formed, they developed the ability to infect the first cells. Could today's single-stranded RNA viruses be the descendants of these peculiar RNA molecules? So still it's a question mark because we don't have anything to prove these theories or this kind of, or say any single theory. So others, they have argued it, right? So some people have argued that precursors of today's of those Mimi virus, they led to the emergence of eukaryotic cells. Then some people described model, some explaining this proposal also. But perhaps both group postulates the current nucleus in the eukaryotic cell, it arose from a endosymbiotic like event in which a complex enveloped DNA virus become permanent residence of an emerging eukaryotic cell. This is they just postulate that. It is, again, it is not not proof, it is not a theory, it is just an hypothesis. So no single hypothesis may be correct. Where viruses came from is not a simple question to answer. One can argue quite convincingly that certain viruses, which are, for example, retroviruses, they arose through progressive process. But what about other viruses? What about DNA viruses? Then mobile genetic elements, they gained ability, transposomes to travel between the cells, becoming infectious agents. Could be, possibility can be there. The one can also argue that large DNA viruses, they arose through regressive process whereby once independent entities, they lost the key genes or a function over a period of a time, and they adopted the parasitic replication strategies. So we cannot say that any particular hypothesis is sure short, but we can still say that okay, these might this might have happened, right? Finally, the idea that viruses gave rise to life as we know it represent is very difficult. Today's viruses, they arose multiple time by multiple mechanisms. Maybe they arose by mechanism which we yet do not know, yet to be discovered, and we still believe that the fields like genomics or structural biology or the NGS like techniques can give certain answers to this, right? So for the origin of viruses, we can say that there are three theories: regressive theory, progressive, and co-evolution of virus first theory. So regressive is like they are degenerate form of intracellular parasites. Progressive means cellular RNA and DNA components or a normal cellular nucleic acids that gain the ability to replicate and then they evolved. Co-evolution means they co-evolve with life. Their co-evolution like with RNA and the DNA molecules when they first formed. So viruses, they evolved with those kind of the molecules along with them, or they keep co-evolved with the other kind of a life on the earth, or they might have evolved much before than that also, right? So at this point also, I mean, we are in 2021, or at this point also, we cannot say uh, clearly and confidently and by saying 100% that any one of this theory is correct. These are just the speculations, these are just the hypothesis, not a theory. So these are just the hypothesis still, and we are still working for it. I mean, there are group of people in the world who are still looking for the origin of the viruses. Their studies or their research is still directed towards the identification of the fact that how viruses have evolved or how they might have evolved, right? So this is still an unanswered question, and we are still working towards this. So this is all for my my side for today's lecture, origin of viruses. This is little tricky, but when you'll study, see, the first hypothesis or progressive is compared there. The comparison is with the retroviruses. The first hypothesis is based on the retrovirus replication. So on that basis, they have said that this might have happened. The second hypothesis is based on the jumping gene or a concept of a transposomes, which are known as jumping genes also. So jumping genes, yes, it can happen it is very I mean it can very likely to happen that jumping genes, when they are moving from one part to the another part of the cells within a certain period of a time, they can acquire some new genes, or they change themselves and they come out as a individual pathogens or individual pathogens which need a host machinery to replicate, because they are totally dependent upon the host, right? So this is how there are still the speculations or hypotheses about the origin of the viruses, and we still cannot say that one particular hypothesis is 100%. Many people are divided in their views and in their thoughts about the origin. Few says that the first one is correct, few says the second one, and few says the third one is correct. So we still don't have any sure shot answer, right? So this is open for discussion also, and after the discussion I'll just give you one assignment question for today's lecture. Just go through these hypotheses, just try to study the on what basis these hypotheses are being given. So it is not difficult, a very simple view on the basis of the present. They have tried to link the the present situation or scenario of the viruses with the past that agar present aisa hai, they are replicating this way, so this might have happened in the past, right? That's it, nothing else. So that's all from my side. If there's any question, it's open for discussion. Otherwise, I'm going to give you one assignment topic for today's lecture.
Ma'am, I have one question.
Yes, Manaswani. Go for it.
Ma'am, so you know how you had mentioned that theory that the RNA material and the RNA viruses were the first, they emerged before cells, right?
Yeah.
So, ma'am, shouldn't that theory be disproved, since how would have the viruses replicated if they didn't have any other cells to, like any other cellular mechanisms to use? Because like even now we know that they enter a
They
host
need
cell and then replicate. Yeah.
they see, that's why, see, Manaswani, that's why I told you that these are just the hypotheses. For every hypothesis, I've not discussed much in detail, but for every hypothesis there is always a point, there's point being mentioned that this cannot be correct. And your point is right, that we cannot say, because see, if we say that they need a living host to multiply or a living cell, to definitely the cell need to come first and then only they can do that, right? So this is the irony of these three hypotheses, that is why not one single hypothesis is correct. So from your point of view, like you said, this is wrong. Yes, maybe 100 different people will come from their point of view that this is wrong. But then there are certain points, like they have said the retroviruses, maybe if RNA were the first to evolve, right? They found RNA before the DNA. They say the RNA is more older than the DNA. So there's a possibility that if, okay, the RNA was there, and see, it could be possible, the possibility could be there that they were like once independent, and then within the period of time they become dependent on the host.
Okay.
So it could be the other way around, right?
Okay. Thank you,
Okay.
ma'am.
Okay, Manaswami. Okay, so one assignment, I'm just trying to create a new assignment, and you can submit that. It will be just one simple question based on today's lecture. Uh-huh. Okay, I have assigned one assignment. It is very simple. I have just asked one question: origin of viruses, your view. So based on today's lecture, you will give your views on origin of viruses, right? So you study today's, it's okay, you, this question you can submit later, because I want you to go through the PPT or go through any material again, and then give your views in against or for or in favor, whatever you want to do. But I want your views. What do you think that this should be correct or it is wrong? If it is correct, why? If it is wrong, why it is wrong? So purely your view. It could be any absurd idea, it could be any nonsense idea which comes to your mind, but only your views. No googling, nothing else. I want to know exactly what your mind works and how your mind works when, if something is being, see, this is, you're in the final year of PSC, so your thought process is very important. Like Manaswani, she just after the PPT, she asked me the question, and this thing was in my mind. I knew, because if you are, listening very carefully, or you're just going deep into it, one person will definitely ask such question. So I want your views. What are your views for this? And what do you think? If it is first theory is correct, then why? If it is not, then why? If second is correct, why? If not, then why? Or third, why? Vice versa. So by evening or by tomorrow, I should get all your answer. I'm not asking for right now, right? So we can go for the attendance also. All of you, please switch on your camera for the attendance. Adhish.
Present,
Yes,
ma'am.
Adhish. Yes. All of you, please switch on your camera. Athoiba.
Present, ma'am.
Ayushi.
Present, ma'am.
Yes. Sam.
Present, ma'am.
Devanshi.
Present, ma'am.
Ishita.
Present, ma'am.
Janvi.
Present, ma'am.
Kshitij. Manaswani. Yes, Manaswani.
Present, ma'am.
Namrita.
Present, ma'am.
Nandini, Nehal,
Present, ma'am.
Rupal,
Present, ma'am.
Sahiba, yes,
Present,
Sahiba,
ma'am.
Sanjana, Shakti,
Present, ma'am.
Present, ma'am.
शक्ति के बाद है शिवानी, शिवानी, तन्वी,
Present, ma'am.
त्रिदिव,
Present, ma'am.
विवेक, युक्ता,
Present, ma'am.
Present,
युक्ता,
ma'am.
युवराज.
Ma'am, present.
Okay. One more thing that I guess the supplementary examination date sheet is also available. So those who have a supplementary examination for even semester, please check your Amazon for your dates. And one more important thing that since examinations are during the time of your classes, so before examination, you will mail your PL, who is Dr. Sunny Dir now, for the OD, for the time period of that examination, so that you don't miss I mean, I don't want you to lose your attendance, right? So make sure that before the exam, not after exam, before exam, you will mail your PL that you have so and so exam from so and so time. For that period, you should be given OD. Otherwise, your attendance will be short and then later cannot be done anything. So please make sure that you do these things, right? Okay, fine. Okay, students. So maybe next week we can see you. Happy weekend. Thank you.
Thank you.
Thank you.
Thank you, ma'am.
Thank you, ma'am.